Okay, hello everyone. How are you today? Looks good. Thank you. So, today we are entering a new switching gear and entering into the whole new entity of life. So, we are focusing uh, in contrast to what we've been up to till now. We are going to look at the life at a very microscopic, small scale level and trying to look inside of the cell so that we can understand each little bit of the structures and their functions. Okay. So I try my best to add as much as possible, add some of the actual content from the textbook onto. So my lecture slide may be a little bit, just a little bit different from what you have as a lecture handout, but overall it's basically same flow. Uh, so anyway, I have to begin with the invention and the development progress of the microscope, because the microscope as a tool like has been instrumental, like literally microscope is instrument, and the progress of the technique in the microscope development has been instrumental in understanding, better understanding of all the inside, the fine structure of the cell. Without it, it would have been impossible at all. Okay, so this we don't really. Who cares about who invented the microscope first or not? But anyway, let's try to give as much credit, due credit to this, the British gentleman, Robert Hooke, who lived a long time ago, obviously. So, yeah, he is claimed to be the first inventor of the first, microscope. But anyway, more importantly, the term cell is the one that he actually coined the word for the first time. Cell, like when he looked all those structure of the cells under his invention, the first microscope, and then those cells, shape of the cell reminded him of small cells, a little compartment of the monarch in British monarch, those priests, small rooms. So that's why he named each little different structure as a cell. And uh, since then, everybody follows that, okay, so this is the cell. Uh, so, like with his own word, the description of what the cells look like is, I took this sentence directly from our textbook. So, and always lagging. I don't know why only this room doesn't synchronize this with my iPad. My iPad shows this, and hey, come on. Okay, so this is actually a replica of what he invented as a first microscope. Uh, looks fantastic, so sophisticated, especially considering the days of his contemporary. And what he also found was, like, he took a sample from pond. Until then, no one actually thought anything would be there, like just the pure water looked like a pond water. So he took several drops of pond water and then placed them under his microscope, and then what a surprise! Like he was able to find so many little creatures moving around. So then, what the and he actually described their movement. So many active and dynamic movements like this. And so is that, which is not really important for us. Now, and we have already went over this. The first cell in the form of a prokaryote actually arose approximately about 3.5 billion years ago. It was long, 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 long time ago, right? Billion years. Can you even imagine how long would that be? Billion years, and the three times of the billion years. Oh my gosh. Okay. And uh, since then, all kind of diversification, as we have seen up until now, okay, under the name of, so to speak, the evolution, generated so many different types of cells, right? Um, and so unicellular, single cell. So this is kind of review what we have seen already before. Some of them are single unicellular organism, and some of them are multicellular. Okay, so those are multicellular organism. So the big difference between multicellular organism and unicellular, always we should not forget that multicellular organism, multicellular, is not merely the number of cells in a body, but differentiation. Okay? Each members in the multicellular organism, each members of the cells constituting that body organism should follow, so to speak, this particular mechanism, what we recognize as what differentiation. Okay. And even more interestingly, some of them are engaged in this symbiotic partnership, living together. We can understand that some of them are living together like as a independent entity. There are lots of different examples we can find. Yeah. One very obvious and famous such example is symbiotic relationship between certain plant and bacteria. Like a plants, group of plants that we know as like legumes, like peanuts and garden peas, and all those related plants don't need any nitrogen-based fertilizer. The reason why they don't need any such nitrogen supply fertilizer is because they establish this symbiotic relationship between this soil bacteria and the plant. And give and take is a give and take relationship. So plant provide the food in the form of sugar as a result of the photosynthesis. And in return, bacteria provide nitrogen, which is really precious source, nitrogen. Everybody needs nitrogen for their protein. That's why we have to eat meat, right? So to speak. So these plants have no sweat in obtaining such a nitrogen source. So that's a very classical, good examples of mutually beneficial symbiotic relationship. But if we look around, there's so many other different such relationships exist. But here, two more extreme cases that we are going to take a look at today is even some of such the symbiotic partnership can exist inside of a single cell. That everybody actually, almost everybody enjoys such internal symbiotic relationship that as we can witness today. So this is the mitochondria. Hey, what is going on here? Anyway, so everybody has mitochondria, right? Like not everybody, but almost everybody. All eukaryotes, all eukaryotic cells have mitochondria. And then if you think about it, where we obtained in the first time originally this mitochondria, the organelle, the theory is trying to explain the origin of this mitochondria existing in every living eukaryote. Strikingly interesting scenario is we call this as an endosymbiosis. I have already explained a little bit about symbiosis, which living together. But endo means what? Inside, internally. What it says is mitochondria are actually captured slaves or captured prokaryote, individually living ones, individually living bacteria. I should be a little bit more correct, like bacterium, a singular. But anyway, the mitochondria was a freely living creature and then somehow captured by bigger guy. And usually the fate of such event would be destruction of this captured one as a food. But for some reason, this mitochondria's life was spared. And then they signed the contract. Okay, you provide me something, and I will provide you what? The protection or anything? Is what this scenario. It's too good to be true, or like even bizarre. How can that happen? Something was captured inside the cell and then still live their life is mutually maintained, intact. But there is no other way to explain the actual origin of this mitochondria because several lines of evidences shows that actually, yes, the mitochondria might be truly something that originally, long time ago, they were living independently, like independent cells, but somehow they came inside of other bigger cell. But anyway, the end result is that now here is the first form of real eukaryotic cell. Once this, whoever they are, they must already have acquired already a nucleus and some other organelles. But one thing is lacking is this mitochondria. What's the function of mitochondria? Anyone? What's the function of a mitochondria in our cell? Yes? Mitochondria? Yes, we have already in the previous lecture that we have seen that mitochondria have their own genes, right? But that is something very interesting feature, but that doesn't say their function. My question is, what's the use of, why do we keep mitochondria inside of our cell? There must be something like the benefit that we can get. Everybody has to contribute something to the entire community, right? So what's the function? Yes? Yeah, the energy is like if we use a metaphor, like mitochondria is something like the energy factory, like power plant. So in the form of what kind of energy do we use? ATP is almost exclusively the cell's energy. And how do we make the ATP? Of course, without mitochondria, because mitochondria was not like the first form of life ever appeared on Earth. So before mitochondria, there must have been some way of generating ATP, because ATP is the universal form of energy for everybody. But the way they generate ATP, I'm talking about the mitochondria, is like another universe. Is they generate this ATP much more efficiently and in much larger quantities. So, like, it's no comparison. Having mitochondria and not having mitochondria, you have no contest. So, if you had mitochondria, then you are able to generate so much huge amount of energy, ATP, and that is the key actually. If you want to become a eukaryote, so like as I went over already some time ago in the lecture, the one big major difference between prokaryote and eukaryote, this is the size. Eukaryote cells are 10 times as large as general average prokaryote cell in diameter. Okay. So with such a huge size, that means you need much more energy. Energy requirement is and how are you going to supply such huge high energy demand? And the mitochondria gave us the solution. In other words, without mitochondria, evolving into eukaryote would not have been possible at all. Is because of this, the really big key breakthrough of how are we going to take care of this, solve the problem of energy supply, especially uh, talking about the multicellularity, it becomes much more clear that in order to become multicellular organism, like another level of, another dimension of this energy requirement comes. So that mitochondria would have been essential, like critical to become. That's why, maybe that's why no true multicellular organism have ever been found in where? In prokaryotic world. Because prokaryotes do not have mitochondria. Like prokaryotes do not have any organelles, right? So including mitochondria. Only eukaryote can enjoy this possession of mitochondria, and what good, what kind of a benefit did we ever get after acquiring this mitochondria? Like this. Energy, so we can become bigger and we can also become multicellular. That's another very two extremely important things. Okay, so we all know that, or maybe we all agree, let's assume, let's pretend that we all agree that all these eukaryotes living today were evolved from originally from prokaryotes. But not every prokaryote, but only some very small particular branch of prokaryotes have succeeded in evolving into eukaryotes, right? Certain branch of prokaryotes during the process of evolutions. Okay. And so eukaryotes, this is another kind of review like type of information. Obviously, eukaryotes have a nucleus and other organelles. All these organelles have in common is this, membrane bound, right? So all these organelles that we define is something that enclosed within their own internal independent membrane. What's membrane? What's the chemistry of a membrane? The same chemistry of this membrane. What? Do you still remember? Phospholipid bilayer. The chemistry of creating membrane is phospholipid. If you still remember the name, the weird name, phospholipid bilayer, which is the material that used to build our cell membrane, and exactly the same such phospholipid bilayer, that membrane, exists inside of the cell and then provide a smaller internal membrane. So chemically speaking, they are exactly identical. This inside membrane is same as the external cell membrane in terms of chemistry. Right? So this membrane-bound internal organelles are the key features of eukaryotes that distinguish them from the prokaryotes. Prokaryotes do not have such membrane-bound organelles, right? So you already have seen this comparison cartoon between prokaryote and eukaryote. All these are membranes, internal membrane, right? Hey. Here, the mitochondria, okay? As we have seen the previous lecture, the number of mitochondria even, not only one or two or three or couple, several, but usually like tens, 20s, or in some cases, depending on different cell type, and some exists in hundreds, depending on, like this is another kind of logically, you can follow this. If any cell in your body, the number of mitochondria differ depending on different type of tissues. Like the tissues that require so much amount of the energy, then the number of mitochondria in a cell tends to be larger, higher. Whereas some cells, I don't need to spend too much energy, then the mitochondrial copy numbers in a cell tends to be smaller. Like so, it follows. It is like correspond to the cell's direct energy demand, which is clever. And several different labeling of organelles in a typical eukaryotic cell, typical animal eukaryotic cells, to scare you, like, hey, there are so many, here are so many things that we need to learn, we need to memorize, right? On the other hand, this is a typical cartoon of a prokaryotic cell. Not much, right? You don't really have too much. You have DNA, obviously, but this DNA is not enclosed with the membrane-bound nucleus. You don't have them. And no other membrane. The only thing they have is so many numerous copies of ribosome. Ribosomes, everybody has to have ribosomes because what's the function of a ribosome? Yeah, exactly. Protein synthesis. So without ribosome, you cannot do anything. So ribosome, every cell should have ribosome, except you don't really have. Obviously, as a prokaryote, they have the prokaryote specific some of the features that we are going to go over later on. Right now, I'm not going to focus any of those details. But anyway, so this terminology is the compartmentalization, which is the hallmark of eukaryotic cell we have already seen even in the exam. And like actually this compartmentalization is directly related with this, the large size of the cell, as we have seen before. These are all such a review type of. So this is also another, why do we need organelles? What's the function of general function of organelle? Unique environment, that's it. Now, let's take a look at first the origin of all these organelles. But one thing is, we have so many different types of organelles there, as we have seen in the previous several previous slide. But there are two different groups in terms of their lineage or origin. There are only two different groups of organelles. One is something called, which is a big word, so I'm afraid you don't, what is my, this is not mine. What is mine? Blue one. Paging my blue marker. Does anyone remember that where I put my marker? Oh, thank you. Another endo thing now. Endo membrane system. So many organelles belong to this group, what we call endomembrane system. They are very similar, not in their functions, but in their origin. So that's why we can safely group them as an endomembrane system, which is big word, but this is nothing but simply meaning endo is what? Inside. So what? Membrane. Everybody knows membrane now. Endomembrane system, a big deal. Already, yeah, organelles are, by definition, it's an endomembrane system. Oh, okay. So what's the excitement? What is the other group? So almost everybody belongs to this big group of endomembrane system, whereas two peculiar organelle belongs to the other group, which doesn't fit into this endomembrane system. But so there has to be something like accounted for or separate, independent in terms of their origin. That is mitochondria. So it cannot go to this endomembrane system for several reasons. It doesn't fit. They are so different. And another such example is very similar to mitochondria, which is, can anyone think of any organelle which is very similar in terms of their structure to mitochondria? But unfortunately, we don't have them. Chloroplast. Right. So chloroplast, only those little green guys out there have this chloroplast. So thanks to this, they can do this magical metabolism we call as a photosynthesis. These two, mitochondria and chloroplast, we cannot put these two into this endomembrane system because they are too different from the rest of things. That's where this so-called endosymbiosis theory had to come to explain the origin of mitochondria and the chloroplast in terms of the evolution. Others, we can try to explain this type of something like internalization of the cell membrane. One clue is, chemically speaking, all those other organelles, the boundary, internal membrane, is exactly identical to the what's there in the cell membrane. That's kind of a safeguard, safe bet that we can assume that, okay, maybe they must have originated from cell membrane, the part of the cell membrane. Okay. What's happening also described in your textbook is this. So this DNA, so this is the ancestor prokaryote that is trying to become a eukaryote, like in the course of evolving into eukaryote. So there is a long way to go for this little poor little guy, because you need first nucleus, right? And you need also some other membrane-bound organelles. How are you going to acquire all such little needed one? And so here is the explanation. So somehow something happened that some pressure from outside pushed some little part of membrane, cell membrane, like this, so that this got folded inwardly, internally. So if you do this, like if you fold somehow this two edge, and then you forgot is fixing this, then it will be later permanently remain inside. That's, I don't know whether this is true or not, but that's probably the best what the scientists can offer as an explanation. So this is, I forgot the name actually. Oh, yeah, there is another big word that I'm not going to even bother to require you to remember. But anyway, this internalization of this membrane, a lot of times it's called invagination. And this actually, the terminology invagination is used in developmental biology when the cell, after the fertilization, when cells develop into different part of our tissues. This is what the process that they have to go through. So folding inside, this all is about the membrane. But anyway, so this, so at least they got now nuclear envelope. And in addition to this nuclear envelope, what they have also acquired is some of other maze-like internal membrane structures. And notice that some area, some part, they are actually connected to the cell, the nuclear membrane. Yeah, doesn't it look like? And there is actually, in our eukaryotic cell, there is a such organelle today that at some point they are connected still with the nuclear membrane. So that at least somehow is valid in accepting this scenario. What is this? Why? Okay. So if you keep doing this, then eventually some of the area got separated and then became independent, different functioning, different function carrying out organelles. Okay, so anyway, there are several different organelles, but all belong to these so-called endomembrane systems, have originated from, in the first place, from this pushing cell membrane internally, is what they are trying to explain. Okay, and putting the origin of this mitochondrial corpus a little bit off, that we are going to take a look at later on today. Let's first take a look at this. The objectives of this chapter is like this. So take a look at more closely, especially the internal structure of the eukaryotic cell, so that we can understand the function. So this is a typical way of studying complex things. So that's reminding us this practice of this reductionism, maybe. So maybe this kind of approach we should avoid altogether to be better, like prepared for whole new complexity of a whole new era. But anyway. So Okay, let's take a look at first the cell theory, which is really boring and dull theory. Everybody knows what these are. So still that we can get a little bit of the significant meaning of this cell theory out of this. So let's take a look at these two gentlemen, German gentlemen, after 100 years after the first invention of microscope. During that period, there has been tremendous progress in techniques in making the microscope. So now, at the time, now it was possible to coin some of the meaningful like theory or information built upon this structure of this cell. So this is called the cell theory, which is uh, cells are the fundamental unit of life. Oh, big deal, of course. All living things are made up of cells. Wow, I didn't know that. And all cells come from. This is actually the third sentence, might be the most important thing. Like in a hindsight, understatement. All cells come from other cells through a process of cell division. In other words, there shouldn't be any kind of the spontaneous generation. So all this living creature must come from through this so-called conception of the biogenesis. Okay, still that one is even no surprise. So these are basically the essence of a cell theory. So in other words, the take-home message out of this cell theory is this probably. So all different cells are related. In what context? What else? Evolution. Sure. So diversity may actually some occasions confuse you otherwise, but the basic essence is even though there are so many like different features, diversity they present or living different organism, but there is some kind of common principles on the underlying all these different cells. So that what we are going to get it after studying. After studying one example of a cell internal structure, so can we obtain some basic common principle of how they work? Is actually the objective of studying this chapter's content. Okay, so there are so many different types of cells in terms of their properties, in physical properties, and obviously their functions. So let's take a look at. Sorry, it miserably failed. Audio is only coming to iPad, so it's no use. Let me just go back and present this probably. Okay, first, outside the cell membrane, Altogether, when you look at this, it looks all fuzzy. It's not really clear what details, what's inside of this cell. This is a typical cartoon, computer-generated image cartoon of a typical animal cell. But if we divide each component, so like we can separate. So a cell membrane, external barrier, and what about the ribosomes? There are numerous copies of ribosome inside of the cell. Okay? Obviously, because all we need all of them because they are so important for protein synthesis. Now, what's endoplasmic reticulum? This is kind of internal tube-like network extended so extensively inside of the cell. And another thing is this is like continuously connected to the nucleus. And this so-called endoplasmic reticulum has a unique function. And another such membrane-bound organelle is a Golgi. It's look like it's a reminiscent of some of the stacks of a pancake in many standard biology textbook. The Golgi apparatus, something, is being portrayed like that, the stack of pancakes. But the real image, more realistic image of Golgi should look like this. And now the mitochondria. As we can see, multiple copies of mitochondria are seen as a typical animal cells. And this is also another, like, in a standard biology textbook, this mitochondria look just fatter and looks a little bit different, but now it's more realistic. Mitochondria looks much slender and the what else? And this slender thing can be also cut into half, and so their shapes can vary depending on different circumstances. Now, that's not all. And some other the other details of organelles are still even missing in this cartoon. And inside of our cell, we do possess some of the skeleton-like structure. This is not organelle. Something called the cytoskeleton is not organelle, but some structural network of proteins. And these proteins are really fibrous, long proteins. And there are so many extensive networks, they are forming the extensive network of these long proteins, and altogether, they provide us with very important function of just like skeleton-like supporting function, as well as some other very important transport function, as we can briefly look at in this chapter 2. And nucleus. One thing, if you didn't know yet, one thing peculiar about this nucleus is this nucleus is also something made up of same chemically identical material as the cell membrane. However, one thing you notice is little pores, small numerous little pores on the surface of this nucleus, through which all this material traffics. So all those material needed inside of this nucleus will enter through each small pores. And also, some of the product made by the activity of a nucleus will also escape through this little pore. So this is really important, the trade point, should I say. Done. Okay. All right. So let's take a look at first the cell membrane. Sometimes it's also called the plasma membrane. So it is the same thing, cell membrane or plasma membrane. One thing we can think about their function. Why do we have this cell membrane? Uh, Obviously, it's a barrier. If we have cell membranes, it excludes some outside the thing and then keep something inside internal things together. So it serves as a really efficient, effective barrier, right? But there is something more to that. Not only mere barrier, but it also provides numerous channels and transporters. What's the difference between channels and transporters? Basically the same. So transporters and channels, maybe you can think channels as a passive passage through the material can move from higher concentration to lower concentration. So it's a natural way of movement. It provides such a whole space. Transporters you can think of as some of the active thing. So even some material won't go such direction from higher to lower concentrated area because there is some discrepancies exist. But still, sometimes even at the spending of the ATP energy, sometimes we need to transport some of the valuable material from outside to inside or vice versa. So maybe let's just take those difference between channels and transport as it is, although it is not entirely accurate distinction. And so this plasma membrane, another thing is universal but distinct. Can anyone guess what that means? This is the characteristic, trying to describe some of the key characteristic feature of cell membrane. This universal but distinct. What does that mean? Yes? So which one? Which one is universal? Which part? Chemistry or existence of the cell membrane is universal? Is that what I mean? Yeah, of course. But also the chemistry, basic chemistry, as a phospholipid bilayer is also very universal. Every different cell, even bacteria and eukaryotic cell and any different kind of cell has exactly basically identical same phospholipid bilayer structure. That's what we can go up to, like universal things. But then what is this? At the same time, this is distinct. If cell membrane, basic chemistry structure of the cell membrane is universal, and how can we achieve each different type of cells achieve this distinction is the question. You have a basically same frame, but then how can you implement the element of distinct features so that we can distinguish this red blood cell from white blood cell in terms of cell membrane? Obviously, of course, their shapes are different. No one can miss it. But if we only focus on the structure of cell membrane, still we can find some difference distinction. But already I have said that chemistry, in terms of chemistry, they are all the same basic phospholipid bilayer, then how can you achieve any element of differentiation? This. So basically, this is the structure, typical structure of the phospholipid bilayer. In every different cell membrane, the cell membrane of every different cell has basically identical chemical structure like this. But still, what you can achieve some distinction is by putting, you still remember the oligonucleotide, oligosaccharide, right? And also, you can put some protein, but this protein have different oligosaccharide. So this type of thing, we call this molecular markers. If you still remember, the function of this carbohydrate, oligosaccharide, as a serving as some recognition marker. Do you still remember what totally washed out of your brain now? That's one of the important function of this little carbohydrate, the oligosaccharide. So with the protein in combination with different type of carbohydrate, we can make some unique membrane environment depending on according to different type of cells. That's what it means in this textbook. Okay, so under this endomembrane system, including endoplasmic reticulum, Golgi apparatus, lysosome, and vesicles are the major type of organelles that constitute the so-called endomembrane system. And overall, what they do, like key characteristic of all these so-called members of endomembrane system is this: import and export of the material. Like import the material from outside into the cell and deliver them into a specific location, and the vice versa. Like some of the materials that we made inside, then transport them into the cell surface or even out of the cell. A lot of cells actually have so many materials to ship out of them. That is, we call as a secretion. Like insulin as a hormone is made in some type of tissue in the cells, and then they are being secreted into the blood. Okay? So how do we transport this insulin made inside of this particular cells, tissue type, and then to which route they are being shipped and how, and then end up the outside of this cell so that they can fly and float into the different area through the bloodstream. It's actually basically hidden message of this. That's why we need to learn a little bit about this, the members of the endomembrane system and what they do. Yeah, this may be a little too much for a lot of you. It's too difficult. So I will try to make this as much as possible to simplify this. But basically, that is the reason why I need to go through these little things. Speaking of, once we have decided to look over the internal structure of the eukaryotic cell for the purpose of understanding their function. And that's something that inevitably we have to go through, although it may be too hard for at least some of you. Okay, so this endoplasmic reticulum looks like. So I will just try to make this as much as possible, as simple as possible. There are two different types of endoplasmic reticulum. All these fuzzy blue lines are the endoplasmic reticulums inside of cell. This is a real photograph. Hey, where are you? So here, this thing is labeled as mitochondria. So this is a little part of the structure. Thanks to the specific type of a microscope called the electron microscope, which is really tremendously high magnifying power. Nowadays, we can take a look at the inside, the little details like this. Even we can view this endoplasmic reticulum like as it is. But here, there are two different kinds of endoplasmic reticulum. By the way, if endoplasmic reticulum, the name is too long, and I don't like this endoplasmic reticulum, the word, the pronunciation. If you didn't like it, okay, then you just simply put them as an ER, which is much better, right? ER stands for endoplasmic reticulum. And there are two different types of endoplasmic reticulum, ER. One is called rough, and the other one is smooth. So if this all endoplasmic reticulum can be shortened into ER, then how would you put this rough endoplasmic reticulum? RER, simple. And what about the smooth? Yeah, they will do. So smooth endoplasmic reticulum, rough endoplasmic reticulum, there are two different kinds. And why we separate them? Because they function differently. Okay, so the rough endoplasmic reticulum is actually engaged in basically transport. Like as I mentioned, as an example, the insulin, like those, the stuff that made inside. And first they ship into this space, the RER, and then go to the next destination and the next destination. On the other hand, the smooth SER function totally different things. Something like detoxification or lipid synthesis, totally different functions. Very interesting, because at first probably they have the same origin, but somehow they got separated into two totally different entity of a function. And nowadays we do have these two different. So this is an example of the function of the smooth endoplasmic reticulum, SER. What does it look like? Our muscle. The reason why I can do this muscle flexing is this whole muscle is connected with our neurons. And once I have to move this cup of coffee to my mouth so that I can enjoy. This is all order come from my brain. And actual execution of this action is my muscle, right? And all these muscle fibers synchronize and then have an action of this contraction, and all is controlled by the calcium. The calcium is have so many different functions, but one of such important function is to have this control over this our muscle contraction, and all those calcium needed for such control of our muscle is stored in smooth endoplasmic reticulum of this muscle cell. So, like detoxification and calcium storage and lipid synthesis are the three major such functions of smooth endoplasmic reticulum. Where are you? Okay, here. Yeah. So this is the very bare minimal summary of the function of smooth endoplasmic reticulum. Majority of those lipids are actually synthesized inside of this endoplasmic reticulum, SER, and detoxification. Anybody has a special plan of do this tonight? And no, for you. Why not? Then you need a lot of help from this smooth ER tonight. All those alcohol you have consumed will be detoxified by the action of actually each cells of you, especially each liver cells. Each liver cells endoplasmic reticulum is the one who take care of this toxic alcohol into something else. And once again, the calcium storage. And speaking of this detoxification, there's one very unfortunate, weird result. Can you read this chemical? Can you try to read this, pronounce this chemical? Yes, benzopyrene. Anybody knows benzopyrene, which is very famous chemical? Benzopyrene, maybe it's pronounced as a benzopyrene, so you're not really kind of okay, what's that? But if I write this, maybe some of you might, oh, that same thing, benzopyrene, which is abundant in where? Cigarette smoke and burnt meat. And so what do you know about this benzopyrene? Cancer, yes, cancer causing. So that's why you are advised against like having burnt meat, and also you are advised against like having this thing, enjoying too much, right? Because you are going to get sooner or later you are going to get cancer, especially in the lung area, right? So this benzopyrene is such a nasty cancer-causing carcinogen chemical. But actually, what happened is the true story is this benzopyrene is really innocent. In a defense for benzopyrene, so this benzopyrene is converted into something else. So we don't really have to pay any attention to this, any details. But this is the name of enzyme we don't have to know. Enzyme will convert, enzyme living inside of this smooth endoplasmic reticulum. Okay. Encounter this as a foreign chemical substance. They will try to convert into something harmless. In the cell, for the sake of a cell, if they encounter any such this like organic, by the way, does it look like hydrophobic or hydrophilic? Why do I have to know this? I don't even know what this kind of line stands for. But try it. Give it a try. What do you think? Is this going to be hydrophobic or hydrophilic? It all depends on whether is it going to be a polar or a non-polar. Is it non-polar, right? So yeah, it is hydrophobic. So kind of a general rule of thumb for us is any foreign substance come in, and if that's especially hydrophobic, then cells trying to convert them into some kind of a polar hydrophilic so that it can. Why? Because hydrophobic molecules are so dangerous because they can penetrate. They can penetrate so easily through cell membrane without any stoppage, any interruption. Hydrophobic molecules have a free passage through cell membrane. So you cannot really track carefully where they are going. But if they are hydrophilic, then at least they cannot really easily pass through anywhere in the cell membrane. That's one such an action for us to sequester them. So that is a general rule of thumb of detoxification usually. Okay, usually. So following that rule, this enzyme work diligently to convert them into something like finally the final product is something like different benzopyrene diol epoxide or whatever, but the difference is some of the oxygen containing polar now, so it must be now something at least a little bit of a hydrophilic, so they cannot really easily pass through and go anywhere. And then they pick up specifically them and then discharge them through our the digestive system, and then let them out of our body system. Okay. But in this case, unfortunately, this benzopyrene diol epoxide is much more dangerous than the original, this benzopyrene. Actually, if we didn't do anything, this benzopyrene will not really do any too much harm. But now what they are doing is this. Once we convert them, detoxify this, it can insert themselves in the middle of this DNA double helical structure easily, because the structure of this converted the substance is very much similar to the complementary base pair between two nucleotides. So they can be easily inserted into the middle of our DNA double helix. And what happens if they do? And during the DNA replication, this enzyme responsible for this DNA replication will have much more higher rate of a mistake, like a skip. A lot of times it will skip this area and then do this, and this skip is something disastrous in terms of the end result of the DNA replication. And what is it? That's by definition is a classical example of a mutation. That's why we label this benzopyrene as a mutagen. That's how this benzopyrene can cause mutation and leading into the cancer. So all cancers are the result of the mutation in our genes, obviously. So once again, this particular case is due to this inadvertent the effort of the detoxification in our cell, and actually end the result is we have created much more harmful chemicals than the original one. Is one very rare exception of this, the overall activity of smooth endoplasmic reticulum detoxification is. Yeah. Okay, so that's about smooth. And what about the rough endoplasmic reticulum? The reason why from the external observers, rough versus smooth, under the electron microscope, one group of ER, endoplasmic reticulum, look something very rough on the surface, whereas the other group of endoplasmic reticulum looks very smooth on the surface. Why? Because this particular something RER, rough endoplasmic reticulum surface, is all covered with ribosomes. Ribosome's function, mind you, what's the function of ribosome? Protein synthesis. And what business of this ribosome there attach it to the surface of endoplasmic reticulum is the question. Wherever you see the ribosome, then there must be something going on in terms of a protein synthesis. Ribosome will make protein. And the question is, what happens to the proteins that made by the ribosome on the surface of the so-called rough endoplasmic reticulum? Is this, okay. All those proteins, once they are finished, they are being actively pushed inside of the rough endoplasmic reticulum. So this is a cartoon. This is a ribosome. And this 1, 2, 3 is a stepwise procedure of this, what happens to the proteins made by this ribosome on the surface of RER. Once the protein is made, and they are being pushed inside of this rough RER. So there is a space, okay, inside. So this is a protein. So what's the difference? Can you tell the difference between number 1 and the number 2 in terms of the protein? You have made a protein, whatever it is. Okay. Let's say this is insulin. You made an insulin protein. Insulin is a hormone, but it is the protein hormone. So let's say this is insulin. So the number 1, the procedure, step 1, you just generated insulin. And now what? Number 2, what happened? What difference? As you see, folded. Okay, good. This protein is now folded. Maybe that is something kind of spontaneously can be done, or maybe some other helper might be involved, but whatever. The end result is now this protein is folded. What's the significance of this protein folded? Like difference between this protein in the stage of number 1 and protein in the stage number 2 in terms of what? Utility. Which one? This one is useless. It's a garbage. Now this protein, whatever it is, if this protein is ever going to function as a real useful insulin, then first it has to be like in a properly folded shape. Protein folding is that important. Function. Protein folding will provide the real function of the protein. Without it, it's a garbage. Actually, some of the very important protein for us, due to a mutation, a very minor mutation, they miserably fail in this stage. They cannot fold into proper form. And then what happened to them? They are all destroyed inside. They never make outside of this ER. So the patient of having such mutation will never have any single protein of that particular one. And that causes really, really serious illness we call as a cystic fibrosis. Cystic fibrosis is Korean for cystic fibrosis. Probably not many of you are familiar with this particular illness because as a Korean, we have a really, really, really rare incidence of this genetic disease. But Caucasians, those white population has a really high incidence of this cystic fibrosis. So that's why every biology textbook actually covers this particular instance. But anyway, so protein folding is very important. Now, another thing is what happens. Is there like protein folding only, but something else is going on over in step 2? What is this? Hmm? Sugar chain. What's sugar chain? Oligo, yes. This is an oligosaccharide. So now, if you've been wondering where all this protein on the membrane get this, whatever this oligosaccharide, and now you have the answer. This, the inside of the endoplasmic reticulum, to be more accurate, the rough endoplasmic reticulum is the place where all these proteins obtain their oligosaccharide first. But this is not the only place. This is the first place for the protein to obtain these sugars, but there is more. So in comparison with, if I compare this with the makeup, cosmetic makeup, probably this is basic foundation makeup, and more cover up, more detailed, fine detailed makeup will be done in the next destination too. But this, what's the importance of this sugar chain? Once again, if you have this particular one, then this one will obtain what? Some marker. So now this one, like somebody else will be able to recognize correctly this. Oh, you are insulin, for example. Okay? That's why all these things, you have another unique pattern of these sugars on the surface of your red blood cells too. Type A, type AB, type B, type of this different type of sugars, all serving as a recognition marker. Okay, as much as I just continue to go on, but it's hit 16, so I have to stop and 25 minutes break. So you are free, at least for now.